Dear students, till now in the chapter Microbes in Human Welfare, we have discussed about the role of microorganisms in household products and the role of microorganisms in industrial products. At industrial scale, we had discussed that the commercialization is going to occur of these products, so they are being made in very large amounts in specific vessels which are known as bioreactors or fermenters. We have so far discussed about two such products obtained at the industrial scale. One of the product was the production of different types of beverages, that is alcoholic beverages, and we had also discussed that what was the raw material used for making those alcoholic beverages. Another thing we had discussed was different types of antibiotics which can be prepared on the industrial scale using microbes. The third products that can be obtained for commercialization and industrial purposes is the use or the formation of chemicals, enzymes and other bioactive molecules. I am repeating again, chemicals, enzymes and other bioactive molecules. So those substances, bioactive molecules, are being used in the pharmaceutical industries for treatment of different diseases. There are certain drugs, certain medicines that are being prepared at the industrial scale. So first, microbes are used for the commercial and the industrial production of three important things. The first one, that is organic acids. The another one, that is alcohol, and the third is the enzymes. So for all the making of these three things, there is use of microorganisms, organic acids, alcohol and enzymes. Let us first talk about the organic acids. Now different types of organic acids are used in the medicine purpose. They are also used in the large scale in the chemistry laboratories for testing, for analysis. So all these organic acids that are produced using different types of microbes. Some are bacteria, some are fungi. So first let us discuss acetic acid. The acetic acid is produced using the microbe Acetobacter aceti. The name of the microorganism is Acetobacter aceti, which is a bacteria. The another one is citric acid. The citric acid is produced using a fungus Aspergillus niger. Aspergillus niger. Lactic acid is produced using Lactobacillus, even in small scale and even in the large scale. Butyric acid is produced using Clostridium butylicum. Clostridium butylicum. So in this way, we have acetic acid, citric acid, lactic acid, butyric acid, which are being produced with the use of different types of microorganisms. Now, let us talk about ethanol. The production of ethanol is being done by the yeast, that is use of yeast Saccharomyces cerevisiae, which was also known as baker's yeast and it was also known as brewer's yeast. Okay, so ethanol will be produced using yeast. The third important product obtained at industrial scale is the different types of enzymes. The first enzyme that we talk is lipases. Lipases are the set of enzymes that are used to remove the oily stains from the clothes. So oily stains from the clothes, from the laundry, can be removed using the lipases, and therefore lipases are added to the detergent formulations. So when the detergents are prepared, lipases are the set of enzymes which are added to remove any type of oily stain from the clothes. The second type of enzymes is pectinase and protease. The pectinase and protease are used for clearing the bottled juices. As we know that when we prepare some juice at home, right, it is not very clear, but the juices that are available in the market, the marketed juices, they are very clear. So for clearing them, for clearing the bottled juices, the enzymes used are pectinases and proteases. This was asked in this year GSEB paper also. So it's an important MCQ. Another thing that can be prepared using enzymes, that is another enzyme is streptokinase. The streptokinase is the enzyme made up of streptococcus. It is made using the streptococcus bacteria, and the usefulness is to remove the blood clots in the blood vessels. So in the blood vessels, the blood gets clotted, and if it has to be treated, the doctor will inject with the mixture of streptokinase. So streptokinase, here we have discussed about 3 enzymes. The first one lipases, pectinases and proteases, and the streptokinase, which is used to remove the blood clots from the vessels. Now another bioactive compound is cyclosporin A. It is cyclosporin A. Very importantly, remember the name of the organism that produces it, Trichoderma polysporum. The name of the organism is Trichoderma polysporum, and it is an immunosuppressive agent. So during an organ transplant, we know that our immune system is very strong and it will immediately react and not accept any foreign blood group, or it will not accept any foreign organ. So for that case, it is important that immunosuppressive agents are given so that your immune system can be suppressed for a short interval of time. And this suppressing of the immune system can be done using the immunosuppressive agent called cyclosporin A. And lastly, remember it was produced by Trichoderma polysporum. And lastly, we would like to say that another bioactive compound is statins. The statins are produced by the yeast Monoscus purpureus. The name of the yeast is Monoscus purpureus, and it is used for lowering the blood cholesterol level. So lowering the blood cholesterol is the function done by statins. The two things are very frequently asked in the exam, cyclosporin A and last statins. These two are very commonly asked. But sometimes MCQs are also being taken from the other topics, like any of the organic acid can be asked, and the name of the microbe which is responsible for producing it can be asked, or it can also be asked about any of the bioactive compound.